Welcome back to Catalyst University. My name is Kevin Tokoph, and in this video and the next few videos, we're going to be taking an in-depth look at steroidogenesis, the biosynthetic pathway by which your body manufactures these steroid hormones. Now, in this specific video, we're going to be looking at the synthesis of cortisol and aldosterone. The next video, we're going to be looking at the synthesis of the sex steroids, androgens and estrogens. So we won't be hitting that in this video, but those molecules ultimately come from some of the molecules that we will see in this video, and we'll kind of bring that out as we get there. So, first thing, very important, a lot of the sex steroids in humans are made in the adrenal glands. Now, the adrenal glands have two general components. There's the medulla, which is the deepest component, right in the dead center. And then outside of the medulla, you have the adrenal cortex. Now, the adrenal cortex is divided into three separate layers. From deepest to most superficial, we have the zona reticularis. There are certainly some sex steroids that are made there. It's not the primary site of sex steroid synthesis. That's actually in the gonads. But there are things like dehydroepiandrosterone that we'll see in the next video and other things like that that are made there. Now, superficial to that, we have the zona fasciculata. This is the area where the vast majority of cortisol is made. And then superficial, that is the zona glomerulosa. The vast majority of aldosterone comes from this region. So just bear that in mind as we go forward. Now, any discussion of steroidogenesis, we have to begin with the parent steroid, the molecule from which all other steroid hormones come from, and that you can see on the left side of the screen right here. That is the molecule cholesterol, this 4-carbon ring organic molecule. Now, a real brief aside, we'll get back to the major stuff in just a minute, but we will see this nomenclature pop up throughout the video. Cholesterol and all of its derivatives have a certain naming system that we use for the carbon rings. So if we start with the carbon ring on the leftmost side, at least the way it's drawn now, that 6-membered ring is called the A ring. As we go from left to right, working towards that 5-membered ring, we have the B ring, the C ring, and of course the 5-membered ring would be the D ring. Okay, just keep that in mind. We'll see that pop up throughout the remainder of this video and the next. Now, depending on several factors like the body's needs at any given time or the cell type in question, whatever it is, cholesterol has many fates, different functions it can perform depending on what's needed and in what location. So some highlights from that. Number one, cholesterol is a constituent of the plasma membrane where it sits amidst the tails of those phospholipids and helps to increase membrane fluidity. Fair enough. Number two, it can be converted into molecules that we call bile acids. This pretty much occurs solely in the liver, and cholesterol can go through a different biosynthetic pathway where it's made into bile acids. These bile acids collectively are stored in bile, which is made in the liver, stored in the gallbladder, and eventually released into the intestines, where it helps to emulsify fats. So it's not cholesterol itself, but it's things that it's turned into, those bile acids that perform that function, but ultimately come from cholesterol. And number three, big important topic here, steroidogenesis. Any cell type, doesn't matter what it is. We could be talking about cells of the gonads, we could be talking about the zona glomerulosa, fasciculata, reticularis, whatever it is, any cell that's going to make a sex steroid hormone has to begin with this enzyme. This is the cholesterol side chain cleavage enzyme, and don't worry, it's the only enzyme in this pathway that has such a long convoluted name, but it essentially converts cholesterol into this molecule called pregnenolone. Now, cholesterol off of the D ring has that large carbon chain we call sometimes the tail. What this enzyme does, as the name suggests, is it cuts off the vast majority of that tail and essentially just leaves a ketone functional group hanging right off the top of that D ring. Okay? And once that happens, you have pregnenolone. Now, important point here, again, any enzyme that I have written in blue, which we haven't seen yet, those are going to be enzymes that are localized to the smooth endoplasmic reticulum. And any enzyme shown in pink is localized to the mitochondria, more specifically the mitochondrial matrix. Now, remember, mitochondria have an outer membrane, and then inside that they have an inner membrane, and then between those two membranes would be the inner membrane space. And then deep to the inner membrane, we have the mitochondrial matrix. And this is where the enzymes exist. So if you look at this picture right here, this actually shows how these enzymes are specifically localized. So they're not just free-floating enzymes in the mitochondrial matrix. They actually have a membranous domain that anchors the enzyme itself to the membrane, and then kind of coming off of the membrane, there's the larger catalytic component that essentially pokes into the matrix, and that's where the catalysis occurs. And the vast majority of these enzymes exist in this way. But in any case, cholesterol is converted to pregnenolone by the cholesterol side chain cleavage enzyme. So more of the pathway now becomes visible, and you see now these enzymes are written in blue. So the pregnenolone that was just made in the mitochondrial matrix now has to leave the mitochondria, go over to the smooth ER, where it can react with either of these two enzymes here, and we can get a variety of products, as you can see. Now, these enzymes' names look fairly complicated, but if you understand the nomenclature a little more, the naming actually makes perfect sense. So let's dissect that by first looking at 3-beta hydroxysteroid dehydrogenase. So in this case, it'll take pregnenolone over to that molecule on the right, which is progesterone, which in and of itself is a sex steroid hormone, also has a wide variety of other functions throughout the body. We'll be talking more about that molecule and its functions in a separate video, so keep an eye out for that. But 3-beta hydroxysteroid dehydrogenase. If we look at pregnenolone on the A ring, that hydroxyl group that's sticking off, so a couple important pieces here. That carbon that the OH group is sticking off, that is the 3 position, thus the name 3-beta. Now, what does beta mean? Well, beta and alpha have to do with stereochemistry and configuration on these carbon rings. So, generally speaking, if the functional group comes off of one of these carbons on a wedge, meaning it's coming out towards you, it gets the designation beta. If it's going into the screen away from you on a dash, it's termed alpha. So this OH group is on a wedge. That means it's 3-beta. Hydroxysteroid dehydrogenase. What this enzyme is going to do, it's going to take that OH group and oxidize it into a carbonyl, more specifically a ketone, which you see in progesterone. And then if we also look in pregnenolone, on that carbon-carbon double bond in the B ring, it's going to essentially move that over into the A ring and create a little bit of resonance between that ketone and that carbon-carbon double bond. Okay? And that is characteristic of progesterone. Now notice here that 3-beta hydroxysteroid dehydrogenase is a reversible reaction. Which way the reaction is going to go just depends on which way equilibrium is pushing it. For example, there's generally going to be a large buildup of pregnenolone that needs to get converted and pushed towards progesterone. So in this case, it's going to push it towards progesterone. But we will see this enzyme later on, actually, in the next video. Then we have 17-alpha hydroxylase. What does the 17 mean? Well, if we look at the D ring on pregnenolone, that carbon of the 5-membered ring where the ketone is coming off, that is the 17 position. So you can imagine this enzyme is going to hydroxylate that position in an alpha configuration, meaning the OH group is going to be on a dash going away from you into the screen. And that's exactly what we see. So when pregnenolone reacts with 17-alpha hydroxylase, you get 17-alpha hydroxypregnenolone. Now, the progesterone that was just formed, if it reacts with 17-alpha hydroxylase, you get 17-alpha hydroxyprogesterone. And then also notice that the 17-alpha hydroxy derivatives at the bottom can also interconvert with each other via that 3-beta hydroxysteroid dehydrogenase. Now, before we go any further, I want to mention one very important thing that's going to lead into the next video. If you look at the bottom of the screen there, those 2 little baby arrows going down from those 17-alpha hydroxy metabolites, that leads into androgen and estrogen synthesis, okay? Like I said, we'll see that in the next video. The reason I mention that is that the enzyme that catalyzes that reaction is called 1720 lyase. 1720 lyase and the 17-alpha hydroxylase are part of the same enzyme complex. So how the enzyme functions depends on a few factors. So right here we see the enzyme in its totality. This is CYP17A1. That's the technical name of this enzyme. It's a P450. And if CYP17A1 is both phosphorylated at the spot that you see on the bottom of the picture, that purple P, and it has an attached cytochrome B5 cofactor, it functions as a 1720 lyase. That's the enzyme that brings you into androgen and estrogen synthesis. If it is not phosphorylated, and if it does not have that attached cytochrome B5 cofactor, it functions only as a 17-alpha hydroxylase. So if you are a cell that synthesizes mainly sex hormones, number one, you want expression of the kinase that phosphorylates that site, and you also want expression of that cytochrome B5 cofactor, because then you'll have the 1720 lyase. Now, not all of the CYP17A1s are going to be phosphorylated and have the cytochrome B5 cofactor, so some of them will be the 17-alpha hydroxylase, and some of them will be the 1720 lyase, but at least you'll have that 1720 lyase. Now, jumping a little bit ahead here, if you look at this bottom row of enzymatic reactions, this leads towards cortisol production, and the one on the top leads towards aldosterone production, just so you know where we're going. If you're a cell type that synthesizes cortisol, so the zona fasciculata of the adrenal cortex, you want CYP17A1 to be expressed because that's the only way you can get 17-alpha hydroxylase. But you do not want that kinase expressed to phosphorylate that site, and you do not want cytochrome B5 expressed, because if they were, then you have the 1720 lyase activity. So they won't have those, but they will certainly have the CYP17A1, so it will function as a 17-alpha hydroxylase, and that will lead you towards cortisol. Okay? And then if you are a cell type that synthesizes aldosterone, so the zona glomerulosa of the cortex, you would not express CYP17A1 at all. Because at the bare minimum, CYP17A1 would function as a 17-alpha hydroxylase, and as you can see, that is an irreversible reaction. Once pregnenolone and progesterone are converted to their 17-alpha hydroxy derivatives, they can no longer be turned back, and you can no longer get aldosterone. Okay? So, bottom line, how different cell types manufacture different steroid hormones depends on the expression, gene expression, of certain enzymes and other proteins involved with the pathway. So hopefully that makes sense. Now let's continue on with cortisol synthesis, and we begin with that 17-alpha hydroxy progesterone. The next enzyme in the pathway, still in the smoothie R, is 21-hydroxylase. Now the 21 position is not on the carbon rings of the steroid. It's actually coming off of that ketone group that's atop the D ring of the molecule. So if we look at that terminal carbon of the ketone off of the D ring, that's your 21 position. And if you look really carefully in the next molecule, 11-deoxycortisol, you can see that it's hydroxylated. So now we've got 11-deoxycortisol. Now, the 11-deoxycortisol then has to leave the smoothie R and go back into the mitochondria, where it's going to react with this last enzyme in the pathway, 11-beta-hydroxylase. Now, where is the 11 position? Well, the 11 position is on the C ring. If you look at the C ring kind of pointing out at like 10 o'clock, so to speak, that's the carbon that gets hydroxylated, and it's in a beta position, so it's sticking out on a wedge toward us, the viewer. Now that we have 11-beta hydroxylated, 11-deoxycortisol, we now have cortisol. And now the cells of the zona fasciculata can give themselves a round of applause, take a break, pat on the back. They've made cortisol, which will eventually be secreted into the blood when it is needed. Now, before we get specifically into aldosterone synthesis, one really important consideration. The body doesn't always need a lot of cortisol, or it doesn't always need a lot of aldosterone. So how does the body know when to start making a lot of it? Well, it has to do with regulation from the brain, ultimately the pituitary. So when the brain recognizes, hey, we need some cortisol, or hey, we need some aldosterone, the pituitary releases ACTH, adrenocorticotropic hormone. ACTH travels in the blood, ultimately in part to the adrenal cortex, where it acts on the cells of the zona fasciculata and zona glomerulosa, and it upregulates these two enzymes. It upregulates 21-hydroxylase. It upregulates 11-beta-hydroxylase. And then those particular cell types can start making their main product, and they make more of it under the control ultimately of ACTH. So if you're ever wondering how ACTH functions, that's in part how it functions. It upregulates those enzymes. So when you have a lot of ACTH, you get a lot of cortisol. Makes sense. Now to the aldosterone synthesis pathway. There we begin with progesterone, and progesterone will not be converted to 17-alpha-hydroxyprogesterone because we don't have expression of the CYP17A1 enzyme and therefore no 17-alpha-hydroxylase. So progesterone, under the control of ACTH with upregulated 21-hydroxylase, will be converted into deoxycorticosterone. And again, you can see here off of that carbon that's sitting on top of the ketone, off of the D ring, it gets hydroxylated and we get deoxycorticosterone. Then deoxycorticosterone leaves the smoothie R, because that's where we've been this whole time, goes back into the mitochondria where it reacts with 11-beta-hydroxylase. And again, you get 11-beta-hydroxylation of that on the C ring, and now you have corticosterone. Interesting note, in some mammals like rodents, corticosterone is actually their main corticosteroid, not cortisol, or at least it's a major contributor, but not in humans. Our major corticosteroid is cortisol. So we're talking about humans mainly in this video. Corticosterone then will react with aldosterone synthase, and you now get aldosterone. And what this enzyme does is it takes that methyl group that's sitting between the C and D rings pointed directly vertical, and it oxidizes it into an aldehyde. The aldehyde functional group, therefore, aldosterone. Now, what input from the body do you need to make a lot of aldosterone? Well, we've already said you need ACTH. That gets you the 21-hydroxylase and 11-beta-hydroxylase. But by itself, that won't do near as much. You also need the input from angiotensin II, another hormone that's going to upregulate aldosterone synthase to get you aldosterone. Remember the RAS system? We've covered that in other videos. Probably learned about it in anatomy and physiology. Renin-angiotensin-aldosterone system. When there's high renin, it stimulates the release of angiotensin II, which then stimulates the production of aldosterone. And the way that it does it is it upregulates this enzyme. And therefore you output more aldosterone. Again, cortisol and aldosterone, their individual functions. We'll be talking about that more in other videos, so keep an eye out for that. But hopefully this video gave you a good understanding about how our body makes aldosterone, cortisol, and also progesterone. In the next video, we're going to follow those little baby arrows down at the bottom of the screen and see how the body makes the sex steroids, androgens and estrogens. And then after that, we're going to look at pheromone synthesis. So it's not technically hormones, but molecules that have effects on other individuals by traveling not in the blood, but through the air. So make sure to join us then. Please make sure to like this video, subscribe to my channel, and hit that notification button for notifications for all videos in the future. Thank you so much.